I got 11 o'clock right now, so if we could go ahead and get started. Our first speaker today is Malin Fex, and the title of her talk is Loss of the Melatonin Receptor Type 1 Results in Dysregulated Alpha Cell Function and Metabolism.
So thank you very much. I would like to thank the organizing committee for inviting me to give this short presentation. Okay, so yes, here they are. Thank you very much. Let's see if I can work this out. Okay, so genome-wide association studies have shown us that there is a common variant mapping to the melatonin receptor 1b locus. This is a receptor that binds the sleep hormone melatonin, and it's associated with impaired risk, with impairment of early insulin secretion and an increased risk of type 2 diabetes. So additional studies were put forth from the same group of people where they also added some animal experimentation, a knockout model of this particular receptor. And if you're interested, there is a recent preprint on bioRxives X where we have tried to model the genetic risk of beta cell dysfunction, just looking at this particular gene and correcting the SNP that was investigated earlier. All right, but now we should talk about alpha cells. But we're staying with melatonin. But just briefly to summarize, from these previous studies, it was shown that the carriers of this SNP, the GG allele or risk allele, they have at baseline higher fasting plasma glucose and also during follow-up, as you can see right here. This is not working, but okay. Also, this SNP is an expression quantitative trait locus, which confers an increased expression of the gene, the melatonin receptor 1b gene, in islets from risk allele carriers. And this is important because melatonin reduces insulin secretion. And the idea is that if you have more of this receptor, you will also get larger dampening of the insulin secretory response over time, which then would confer the increased risk. Okay, so melatonin, as I just mentioned, is a sleep regulator. It is produced in the pineal gland and secreted during nighttime because light inhibits its synthesis. Melatonin does a lot of things in the body, but we will focus a bit on the metabolic part because it plays a role in regulation of metabolic both over time and in a diurnal cycle. The two receptors that bind melatonin are termed melatonin receptor 1b and melatonin receptor 1a. These are both G-protein coupled receptors that suggestedly reduce cyclic AMP, but these receptors can be promiscuous, so there may also be additional pathways. Importantly, they are present in the endocrine pancreas, and the 1b receptor influences insulin secretion, as I said before. The 1a receptor, we don't know that much about, but it is present on the alpha cells as well, and it may influence glucagon secretion. So a number of years ago, some colleagues of mine looked at this in mice, and what they could see from the distribution of the 1A receptor and the 1B, these are termed MT1 in rodents, which is the 1A receptor, and MT2, which is the 1B receptor equivalent, was that the MT2 receptor is predominantly localized to, let's see if this works again, no it doesn't, to beta cells. It's in the H at the bottom, where you can see the co-localization, and the MT1 is mostly present in the alpha cells, as you can see in the D on the upper panel. So, glucagon secretion is fundamentally different from insulin secretion, as we've heard previously. To me, being a beta cell scientist, it's therefore very complicated to study alpha cell function, but also very interesting. So I adopted this image from Gao et al., Journal of Endocrinology, because I think it really shows how complicated glucagon secretion is and what it depends on for actually functioning properly. So, for instance, as we heard before, glucose metabolism is probably more or less necessary for shutting down glucagon secretion because they secrete glucagon at low glucose. But other aspects of glucagon secretion also involve, for instance, glutamate, which is secreted by the alpha cells themselves, cyclic AMP elevating agents or neuronal input that potentially elevate cyclic AMP, also influence glucagon secretion positively, as well as neuronal factors and nutrients, for instance, amino acids. So inhibiting factors could be KTP channel closure. This has also been shown in studies modulating KTP channel activity. Insulin and zinc together also inhibits glucagon secretion. Somatostatin, of course, from the delta cells, and serotonin from beta cells that also negatively influences the secretion of glucagon from alpha cells. So what about melatonin then? Does melatonin play a role in this situation? From a physiological standpoint, it makes sense because it already modulates insulin secretion, so why wouldn't it influence glucagon secretion, right? So we wanted to take a closer look into this. So we utilized a full body knockout mouse. This mouse is on a C3 HE background, and this is also important because most inbred mouse strains have a mutation in one of the enzymes that are critical for the biosynthesis of melatonin. But this mouse does not, so it has intact melatonin signaling, secretion from the pineal gland, and so on. So, because we were interested in glucagon, we started to do batch incubations of islets to look at glucagon secretion. And as you can see, the white bars are the wild type, and the black bars are the knockout. So we do have a reduction of glucagon secretion in response to elevated levels of glucose in the wild type, while in the knockout, you rather see an increase, which actually also sort of looks like the increase in glucagon secretion observed in some type 2 diabetes patients that we discussed so earlier. And then we stimulated these islets with melatonin because there is an overlap between these receptors. So basically you could still have the MT2 receptor in the alpha cells, and we wanted to see if melatonin actually did anything, if it could inhibit this hypersecretion of glucagon. In the wild types, there is a steady reduction in glucagon secretion, and the combination of melatonin actually appears to reduce glucagon secretion even more. However, it did not do that much for the knockout mouse, or the islets from the knockout mouse in this context, so it's still sort of all over the place. The glucagon secretion cannot be inhibited by elevated levels of glucose. Importantly, glucagon content was also increased in these islets. So then we moved on to stimulate these islets with some different compounds. One of those were arginine, which is mostly known as a depolarizing agent, but potentially could also be metabolized within the cell, of course. And what we see here in the black bars is that this treatment appears to normalize glucagon secretion from the knockout islets. Similarly, we see a similar pattern with the addition of high potassium. I would love to be able to use this to sort of show you what I'm talking about. But basically, there is a similar situation where you actually more or less normalize the secretion with a depolarizing agent. So this led us to believe that, okay, is there something going on with the plasma membrane potential, or at least this is one of the things that we wanted to look at. So to do that, we wanted to create a mouse model where we could visualize the alpha cells. So what we did was that we crossed our knockout mouse with a glucovenous mouse. This is a transgenic mouse that has fluorescently labeled alpha cells with a venous protein. So it's driven by the glucagon promoter. And we performed this cross And eventually we got a mouse that was a knockout of the MT1 receptor, but also had fluorescently labeled alpha cells, as you can see right here. So then we measured plasma membrane potential using a plasma membrane potential indicator, as you can see right here, where the red trace is the knockout and the black is the wild type, and then it was quantified also over here. And I wanted to look more closely into the gap between the 6 millimolar and the 16.7 millimolar, if you can see it, because I see the numbers are very small, but it's like the second set of graphs in the, in the bigger graph. There is a tendency towards a difference there, at least. It is not statistically significant at this point, but we need to address this further and make a lot more measurements here, I think. However, I don't think that this is the full story of what's going on in this particular mouse model. So, having looked at glucagon secretion in vitro, we also wanted to look at insulin secretion, of course, because, I mean, we already talked about the paracrine and autocrine regulation within the islet. However, we could not see any differences in vitro insulin secretion in this mouse model between the wild type and the knockout, so they were very similar. However, insulin content was also elevated in this model, so they have elevated glucagon content and also elevated insulin content. Oops, sorry. Then we wanted to look at the in vivo situation, because of course the islet function in a static incubation is over 1 hour, but what goes on in vivo would potentially reflect a more dynamic situation. So we performed an intravenous glucose tolerance test and looked at insulin initially, and as you can see, the black boxes, the knockout mice are slightly hyperinsulinemic, and this is also visible, shown in the second graph as area under the curve. However, glucose tolerance was maintained all the time in the knockout mice, so it appears that they still have normal glucose tolerance, but they hypersecrete insulin in response to a bolus injection of glucose. So we also measured glucagon in these samples, as you can see right here, and there doesn't appear to be much of a difference between the wild type and the knockout mice in this context, as shown as the area under the curve. However, if we look at the individual time points, it's evident that basal glucagon secretion is elevated, so that's the 0 time point basically, and this is then maintained during the whole IVGTT and still present at 75 minutes. So I think that I have convincingly shown you, more or less, that loss of melatonin receptor 1 results in an inability of elevated glucose levels to shut down glucagon secretion in vitro, but insulin secretion is not influenced. Hypersecretion of glucagon can be normalized by different depolarizing agents, such as arginine and high potassium. The PAMPI measurements, the plasma membrane measurements, indicate small differences in the plasma membrane potential, especially between 6 and 16.7 millimolar. But this is not significant at this point, but we're hoping to sort of add more N's to this. Total islet insulin and glucagon levels are increased in the knockout islets, and basal in vivo glucagon levels are elevated in knockout islets. And they are also hyperinsulinemic, but with normal glucose tolerance in response to an intravenous glucose tolerance test. So, going back to this intricate and complicated model, can we find a spot for melatonin in this context? I don't know, but I think that we still have some outstanding questions left to sort of focus on before we can draw any real conclusions. So I think that we need to take a closer look at cyclic AMP and also calcium changes and flux in the alpha cells of these animals. And potentially also go back to KATP channel activity and look more closely into that. Perhaps we need a different model to do that. This is a global knockout, so I mean, it's not, you know, it lacks this receptor all over the body, but potentially a alpha cell specific knockout would suffice in this context, or a more humanized model. And with that, I would like to thank you for listening, and the people who did this work are on the top, and my group and my funding agencies. Thank you very much.
Thank you very much. Thank you to the organizers for inviting me. And a big apologize before I talk. The first word about data is that I'm talking about the wrong disease and the wrong cells. I'm going to talk about delta cells and compare delta cells in human normal non-diabetic donors with those in type 2 diabetes. But I hope it's interesting anyway, because there's not much known about delta cells. All right, so delta cells are in control of islet function because they block alpha cells and they block beta cells, and it's perhaps not quite clear why this is so. There's probably some aspect of additional control or protective mechanism. We showed earlier, and others have also shown us, that alpha cells need somatostatin to correctly respond to high glucose. There's also some evidence that somatostatin release sets the glucose set point. So it's interesting to look at this. Then we found somatostatin resistance in type 2 diabetes in alpha cells, and we believe this is the same in beta and delta cells of humans. So we found that, or the idea was perhaps there is too much somatostatin around if it's constantly being secreted, and that then leads to common desensitization mechanisms by uptake of the receptor. And we'll come back to this also for the delta cell. So how is delta cell excitosis regulated? There is most likely intrinsic glucose dependence, but it's perhaps not quite clear either, because it's hard to look at single delta cells and in absence of beta cells, and they're electrically coupled to the beta cells. So we try to do this in this work here, and then of course we're interested in hormonal power and autocrine regulation and changes in type 2 diabetes. Unfortunately, we couldn't look at type 1, which we would love to do. One slide back. So we look at glucose effect, we look at GABA, GLP-1, or glucagon, adrenaline, and somatostatin. Islets have very few delta cells, so it makes them rather difficult to work with. We use dispersed human islets, cadaveric human islets, and then we mark the delta cells by expressing from a somatostatin promoter a GFP that is expressed in the granules. So we can, at the same time, measure excitosis from these granules and identify the cells. And we use live cell turf imaging and a little bit of patch clamping to understand what these cells are doing. And then we compare, as I said already, type 2 and non-diabetic donors. The project is headed by Omar Himadi, who has now his own position at the Swedish, what do they call it? SLU. It's one of the Swedish universities. All right. Excitosis is measured by turf microscopy. We have done this for many, many years. So here you see one example of a cell that expresses this granule marker, and then you see sort of a film of one of these excitosis events. So the granule marker disappears suddenly, and we count these events. We can then get time course and frequency of excitosis. We can look at other granule behavior, which we didn't do so much in this case, or at least I don't show it because it wasn't that interesting. But we could look at when does the vesicle arrive at the docking site, how long does it sit there, and those kind of questions. So the first question we had was, how are these cells responding to glucose? And so we did this in a paradigm where we do not stimulate with anything else but glucose. So they're completely free. They're there's no patch pipette on them, so we can just look what they do. And then count exocytosis events, and you see there that they're clearly responding to glucose. And the non-diabetics, perhaps a little worse than the type 2 diabetics. I take this with a grain of salt because I'm not sure this is really significant or true, because we have relatively few observations of this due to the difficulties of doing these experiments. But these are from 10 donors each, or sorry, so 10 and 5 donors. So it is relatively clear from these donors that there is glucose dependence at least. Glucose, so this is the other paradigm we have, where we stimulate instead by depolarizing with potassium, and this is done in the presence of diazoxide, so that the cells are really silent before we stimulate. And so this is more akin to the voltage clamp depolarization in patch clamp experiments. So here you also see that there's an increase with glucose, but it's now relatively small. And this is because now there is all the electrophysiology is sort of cut out of the equation, and the only thing that's left is exocytosis, the exocytosis machinery. And so there's a slight effect of glucose, so they secrete a little bit more in high glucose than in low glucose, and this is lost in type 2 diabetes. We can see this by capacitance. If you don't believe me, well, oops. I apologize for this. There we go. All right, we can also see this by capacitance. Basically, there's a bit of a decrease of excitosis in type 2 diabetes. And remember that in the first experiments, this looked a little different, but remember these cells are now clamped, and so all the electrophysiology is cut out. So the excitosis capacity, perhaps you could call it, is slightly worse in type 2 diabetes. So then we looked at somatostatin, because somatostatin is, of course, released by these excitosis events, and we want to know if this had any autocrine effect. So the first experiment was to simply incubate the cells in somatostatin and see how much excitosis would be affected by this. And so if you look on the left side, I'll just describe. So the ND is the non-diabetics, and the plus SST is a really stark response. So they are really sensitive to somatostatin themselves. And this is lost in type 2 diabetes, as you see there. This is a little bit more moderated in 1 millimeter glucose, where there's still a slight effect of the somatostatin, but in general, they secrete so much less that perhaps this is not so relevant. Right. This doesn't quite work here. There we go. So this is current clamping, so we can measure the membrane potential of these cells. And this is only done in non-diabetic cells. So if we incubate, or if we puff on somatostatin for a very short moment here, as 20 seconds of somatostatin in each of these puffs, there's clearly a fast response to somatostatin. So there are two effects. One is this direct effect on the excitosis machinery, and the other one is on the electrophysiology of the cells. We also checked, so if they're out-inhibited, then you might think that if you incubate with a blocker of the somatostatin receptor, and we tested here a blocker for SSTR2, that they might release more somatostatin, or have more excitosis, and this is really what we see here. If you compare the 10G there with the 10G plus the blocker SYN, then there's more excitosis in those conditions. Also in the free-floating, non-clamped paradigm where we just look at glucose-stimulated excitosis, we see this effect that there's more excitosis with the SSTR inhibitor. Right. So so then we wondered why there might be a resistance to somatostatin in type 2 diabetes. So we looked at expression of the somatostatin receptor 2 by staining of sections in non-diabetic and in type 2 diabetics, and we feel at least there is both less expressed in the type 2 diabetics compared with the non-diabetics, and also more internalized somatostatin receptor. And this is very similar to what we saw in alpha cells, which are also resistant to somatostatin in type 2. Right. This is perhaps interesting for this audience also. So adrenaline inhibits excitosis in the non-diabetics, as you see there in the first slide. At 10 glucose, the effect is pretty strong. But even at 1 glucose, it's clearly there. So those are the gray and dark gray traces. And then if you look in type 2 diabetes, it's reversed. In particular, at low glucose, it's reversed, so that adrenaline now stimulates. And that came, of course, as a surprise. Adrenaline also blocks membrane potential activity, as you see down there in D, very fast, so it's probably a G protein coupled channel that's activated there. Insulin has relatively little effect. Perhaps there's some at 10, as you see there, but this is not significant. But not much happens with insulin in these cells, so they're incubated in insulin. The responses are relatively similar to what we see without insulin. GABA has a strong stimulatory effect, which you see there perhaps the best in the very first, again, if you compare ND and then ND plus GABA, very strong increase in excitosis. Perhaps a little less in the type 2 diabetics. And it's the same at both low and high glucose. This is also both an effect on the excitosis machinery, but also an effect on channels, because we see an increase in electrical activity when we puff on GABA onto these cells, which you see there in G. So finally here is GLP-1, and this is very similar to the responses to, I should show you both. I'll show you glucagon and exendin-4 as a GLP-1 receptor agonist. And we see stimulation by both of these, and by phoscolin also, which I don't show you. So cyclic AMP increases excitosis in these cells, and it doesn't matter if it's high or low glucose or if it's a diabetic cell or not. It will always stimulate quite strongly. This is it. So in summary, what we learned about delta cell regulation is that delta cell excitosis is slightly reduced in type 2 diabetes, and this is similar to what we found in beta and alpha cells. In type 2 diabetes, the excitosis machinery works a little less well. We find that somatostatin and adrenaline inhibit delta cells, and this is an effect both on excitosis and the electrical activity. And curiously, it's reversed in type 2 diabetes for the adrenaline, at least. And it's lost in type 2 diabetes for somatostatin. So somehow this inhibitory effect of adrenaline and somatostatin is lost in type 2 diabetes. And we had some idea of how this works mechanistically, at least for the somatostatin receptor 2. And then finally, I think this was not so clear before either, GABA stimulates delta cell excitosis, both via excitosis directly and via the electrical activity. All right, and these are the people that did the work. This is mostly done by Omar Yamadi, and then with other people from our lab. And we got audits from Edmonton and Uppsala. So thank you very much for listening.
Okay, so our third and final speaker is Rui Gao from the University of Oxford in England, and the title of her talk is Disruption of Electrical Coupling Between Beta and Delta Cells Results in Impaired Glucagon Response to Hypoglycemia in Type 1 Diabetes.
Thank you very much for the introduction. So I'm Rui, currently a clinical research fellow and a postdoc researcher in Petra Rosman lab. I'm honored to have this opportunity to present some of the studies that we did on electrical coupling between beta and delta cells, which may shed light on the impaired glucose counterregulation in type 1 diabetes. The complex figure I put here on the left summarizes our findings, but I will explain this to you in the next 15 minutes. So we all know that diabetes is a high glucose problem, but it's also a problem of low glucose. So hypoglycemia, or having too little sugar in the bloodstream, is one of the major concerns for diabetic patients, and also a significant barrier for diabetes treatment. hypoglycemia can result in coma, reduced hypoawareness, and increased mortality, and which can account for up to 10% of deaths in people with type 1 diabetes. But how does hypoglycemia occur in type 1 diabetes? So the preliminary data from our LEGEND-D clinical trial showed that during the insulin-induced drop in plasma glucose during a hyperinsulinemic and hypoglycemic clamp showed that the drop in the plasma glucose could trigger around 7-fold increase in glucose secretion in non-diabetic controls. However, this response is impaired in people with type 1 diabetes, and this could further contribute to the impaired glucagon counterregulation and hypoglycemia. So glucagon is secreted from the pancreatic alpha cells, but how these alpha cells become dysregulated still remains unknown. So alpha cells are subjected to a complex interplay of various factors, including intrasync glucose sensing, sympathetic neuronal modulation, and a paracrine interaction with the neighboring beta or delta cells within the pancreatic islets. However, the very limited access to type 1 diabetic islets is the major challenge in investigating the underlying mechanisms. So to overcome this, we use NOD mice, a polygenetic mouse model of human type 1. So NOD mice develop diabetes as they age. In our studies, experiments were conducted in young non-diabetic NOD, adult diabetic NOD, and their age-matched non-diabetic controls. So the pancreatic islets pathology in these mice showed a similar characteristics as in type 1 diabetic patients with immune cell infiltrations. So we first look at the NOD mice glucose secretion in response to hypoglycemia in vivo. So in non-diabetic NOD, lowering the blood glucose using insulin could elevate the plasma glucagon by around 4-fold. And however, in type 1 diabetic NOD, the effect was abolished and showing the same pattern as in type 1 diabetic patients. And this would suggest that NOD mice is an appropriate model to study the mechanisms of defective glucagon counterregulation in type 1 diabetes. And we first wonder whether this impaired secretion in NOD mice, is it due to the defective release machinery in alpha cells? So arginines could stimulate glucagon secretion via selectrogenic entry. And we found in the perfused pancreases in non-diabetic and diabetic NOD, the effect of arginine is identical. So this would suggest it is not because the alpha cell lacked the capacity to produce or release hormone. In type 1 diabetes, due to the loss of beta cells, there's an increased contact between pancreatic alpha cells and delta cells. And the somatostatin secreted by delta cells is a very potent inhibitor for glucagon secretion. And we then wonder whether it is possible that the delta cell paracrine inhibition of alpha cells is enhanced in the context of type 1 diabetes. So using the islets, NOD mice islets, we found as the disease progresses, the glucagon secretion becomes gradually impaired. And if we measure the somatostatin secretion in 3 types of islets under the same condition, as you can see, it is significantly elevated in adult NOD mice. And if we block the somatostatin receptor signaling using an antagonist, CYN154806, as you can see, the glucagon secretion in diabetic NOD mice can be elevated to the same level as in the control. And in vivo, during the insulin-induced hypoglycemia, the failure of low glucose to elevate plasma glucagon can be recovered with the application of a somatostatin receptor blocker, CYN, in diabetic NOD mice. So this would suggest that it is the reduced glucagon secretion at low glucose that is caused by the increased somatostatin release in NOD mice. So we also test this using human islets. So in islets from the non-diabetic donors, reducing the glucose from 6 to 1 millimolar could increase the somatostatin secretion. However, the stimulatory response is absent in islets from the type 1 diabetic donors. And like in the NOD mice, the somatostatin secretion is dramatically elevated at 1 millimolar glucose in type 1 diabetic islets. And the application of a somatostatin receptor blocker, CYN, could significantly increase the glucose secretion. So this would suggest there is indeed an enhanced delta cell paracrine impact. Then the question might arise why somatostatin secretion is increased at low glucose in the context of type 1 diabetes. So if we plot all the NOD mice islets, the secretion with x-axis showing the insulin content and y-axis show the insulin and glucose secretion, as you can see, as the insulin content level gradually decreases, the somatostatin level, sorry, it's not working very well, the controls. So the somatostatin secretion becomes gradually elevated, whereas the glucose secretion are significantly reduced as a consequence. And as type 1 diabetes is an autoimmune disease that most characterized by beta cell destruction, and we wonder whether this enhanced somatostatin secretion at low glucose might be due to the loss of beta cells. So our previous study showed that the delta cells are electrically coupled to beta cells via gap junction formed by connexin 36. So at high glucose, the beta cell KTP channel activity is low, and this leads to membrane depolarization. And this depolarization can spread to the neighboring delta cells via gap junction, and thereby controlling the delta cell's electrical activity. Then what about at low glucose? So the electrical coupling also allows beta cell to share their repolarization with delta cells. So we hypothesize that at low glucose, the beta cell KTP channel activity is high, and this leads to membrane repolarization. And in this scenario, the gap junction function as an electric brake, allowing the beta cells to share their repolarization with delta cells. And as a consequence, this further leads to the suppression of action potential firing and reduction in somatostatin release. So to test this point, we generate an optogenetic mouse model that has a halorhodopsin exclusively expressed in the pancreatic islets. So halorhodopsin is a chloride channel that can be opened in response to red light. So we simultaneously patch clamped a pair of neighboring beta and delta cells within the islets. So the photoactivation of beta cells could induce an outward current, trigger hyperpolarization, and inhibit action potential firing at 10 millimolar glucose. And very interestingly, in delta cells, although that they do not express halorhodopsin, we also recorded an outward current, hyperpolarization, and suppression of action potential firing. And this would suggest that the hyperpolarization of beta cells can spread to the neighboring delta cells via gap junction. And if we remove this electric brake using a connexin 36 gap junction blocker, mefloquine, well, as you can see, both in the mouse and human islets, the basal somatostatin secretion increases, and this further leads to the decrease in glucose secretion at 1 millimolar glucose. And this replicates the islet hormone secretory defect that we see in type 1 diabetic islets. And in type 1 diabetes, the beta cell death could disrupt the interaction, the beta to delta cell electrical coupling. And we wonder whether in this case, mefloquine was still showing an effect. So using the islet from the NOD mice and type 1 diabetic donors, we showed that mefloquine had no effect on either somatostatin or glucose secretion. And this would suggest that beta cell loss or the reduction in beta to delta cell electrical coupling may indeed underline the hypersecretion of somatostatin at low glucose. So what we can say so far is that in physiological state, the beta to delta cell electrical coupling is important for maintaining the appropriate amount of somatostatin and a normal paracrine tone on alpha cells at low glucose. However, in type 1 diabetes, during hypoglycemia, the destruction of beta cells could reduce the connectivities of gap junctions between beta and delta cells, and therefore could remove this hyperpolarizing influence of beta cells onto delta cells. And this could further lead to the hypersecretion of somatostatin and impaired glucagon counterregulation in the context of type 1 diabetes. And then what's the clinical implication in the context of type 1 diabetes? We wondered how much beta cell destruction is required to increase the risk of hypoglycemia. So using mathematical modeling, we simulate the beta cell deletion within the islets, and we found that the beta to delta cell connectivity only start to steeply decrease when 90% of the beta cells were deleted. And this is actually in a pretty good agreement with the clinical observation that the hypoadmission risk only start to steeply decrease when 90% of the beta cells were destructed. When C-peptide level dropped below 30 picomole. And that is around 10% of fasting C-peptide in a healthy individual. So in other words, 90% of beta cell destruction. So this would probably suggest that in type 1 diabetes, the protective effect of the residual beta cell function in reducing the risk of hypoglycemia is mediated by beta to delta cell electrical coupling. And this also aligns really well from the preliminary findings from the LEGEND-D studies. So if we characterize the type 1 diabetic patients according to their remaining beta cell function, so those with C-peptide more than 30 picomole and those less than 30 picomole, and we found that during the insulin-induced hypoglycemic phase, during the clamp, the type 1 diabetic patient that was a better remaining beta cell function, so those C-peptide more than 30 picomole, would show the relatively better glucagon release in response to hypoglycemia. And as regard to the clinical implication of a somatostatin receptor 2 blocker in preventing the hypoglycemic risk, I will leave this excitement for Richard from Ducara Therapeutics to present this afternoon about their latest findings on the clinical trials. So in conclusion, in type 1 diabetes, the excessive somatostatin secretion contributes to the impairment of counterregulatory glucagon secretion. And the beta cell hyperpolarizing at low glucose may spread into this neighboring delta cell via gap junction and function as electric brake. And as beta cells are destroyed during the disease progression of type 1 diabetes, delta cells lost beta cell inhibitory regulation via gap junction and therefore resulting in an uncontrolled somatostatin secretion. And that's all for my talk. I would like to thank all the colleagues for their great contribution in this study, as well as our founding bodies. Thank you very much for your time, and any questions are welcomed.
All right, let's give another round of applause for our speakers.